Thank you for taking the time to join us here today. There's something on here that I need to click. Got it. Okay. I hope that you will find it worthwhile. What I hope you to get today after you leave after the talk is that to have ideas in your mind of how you can incorporate or leverage the services that we provide at iCEM and incorporate the power of Cryo-EM into your research. And as you listen to the talk, keep in mind that as different capability of Cryo-EM is being presented to you, keep in mind that all of those are available to you. Even though currently in our facility, we only have one instrument, but we are reaching out to other facilities within driving distance, at institutions within driving distance, to bring you the full range of Cryo-EM capabilities and state-of-the-art instrumentation. So as you listen to Natalia talking about different capabilities, let your dreams run wild, and then talk to us and see if we can make those dreams come true. Okay, that sounds a little bit like an infomercial for Disneyland, but we used to take our kids when they were younger to Disneyland. Maybe I'm a little bit biased, but our Cryo-EM facility is better than Disneyland. I mean, we actually do make science dreams come true. So I'm grateful to have Dr. Natalia Livar here with us to share with us the current state of Cryo-EM. And thank you for doing this, and the floor is all yours.
Of course. So thank you everyone for being here, and thank you, Gwen, for your amazing introduction. So as Gwen explained, I would like to explain to you not only all technology advances that you will be able to get with the Glacios you have currently here, but as well as the new things, new toys that you can add into that Glacios if you want at some point, for different applications. So I am Natalia. I joined Thermo 2 years ago, a bit more than 2 years ago, as a senior scientist. Before that, I was working as the director of 2 electron microscopy laboratories, one doing all the classical clinical pathology samples, the resin, embedded organoids, cells, tissues, all that stuff, and the other lab was all cryo-imaging, high resolution, so single particle, tomography, microelectron diffraction. So I was there for almost 5 years in NCI, and after I decided to move with Thermo. I have over, I don't want to think about how old I am already, I am, but I have almost more than 16, almost 17 years of experience working with electron microscopy, going from traditional TEM to cryo, as well as tomography, etc. So if you have any questions, please, I want this to be very interactive and not me talking for over an hour. So feel free to interrupt me whenever you have a question, okay? And similar for the guys virtually, please. Feel free to unmute yourselves and ask the question. So with that being said, I don't think I need to spend so much time here because the audience here, everyone knows about this, but just in case to put everyone on the same page and to highlight how important all the techniques are and how they are overlapping to each other. You can see, for example, in green, you have the light microscopy, super resolution, conventional SEM, TEM. Like they are very good techniques to be able to analyze very large field of view, but they don't give you the resolution you want. On the other side of the spectrum, in purple, you have NMR and crystallography. Like they are quite of the opposite. They can give you a very high resolution, but they don't provide you with a large field of view. And, okay, I am a bit biased here because I love electron microscopy, even if I was a crystallographer during my PhD. So I transitioned to the dark side with electron microscopy. But I love it. Why? Because you have a balance in between being able to have a very high resolution and being still able to have the large field of view. That being said, I like to use all the techniques here. Why? Because to really understand your biological problem, you really need to use all the techniques you can under the sun. So it's like kind of the structural biology toolbox. The more you use, the more information you will get from your biological system. So again, these three are the main, structural biology techniques. You have X-ray, you have NMR and cryo-EM, and you can see, X-ray is still very high. Plenty of people are using X-ray and it's working very well, and it's very useful technique. NMR is kind of getting a plateau in right now, and you can see we estimate like cryo-EM is going to be exploding probably in the next 5 years or even less than that, and it's exploding already right now, to be honest. Not only because of the software and hardware, but because all the applications we can get with cryo-EM and all the results we can get. Again, I am a guy, I like to use as many techniques as possible. And as I said before, a structural biology toolbox. Sometimes cryo-EM is not the way to go. Sometimes you need to do crystallography, or sometimes NMR, or sometimes cryo-EM is the way to go. And if you can combine all the three, even better with mass spectrometry sometimes, that's the way to do it, guys. So all the techniques combined together will give you the real answer you need. So if we dig a bit more into cryo-EM, so everyone here already knows, cryo-EM has three main techniques. So of course, single particle analysis, cryotomography, and microelectron diffraction. So I will explain, applications and results coming from all the three. The main difference in between single particle, cryo-ET, and micro-ED, first of all, single particle is high resolution. We are getting almost near atomic resolutions right now with cryo single particle analysis, but it's still ex situ. So you still need to express and purify your protein or micromolecular complex of interest, put that in a grid, take data in the microscope, okay? So that's very cool, but what happens if you really want to see that protein in the context of the cell or the tissue? In that case, you need to go to cryotomography. And that is a very good technique. Even the resolutions right now, they are not as high as with single particle analysis, but because you can realize and you can understand the sociology of your protein in the context of the cell and tissue. That's something can be done in your Glacios here, but you will need an extra, kind of toy to add in that Glacios, it's called the selective energy filter. And I will explain later how it works. And microelectron diffraction is very cool because you can really get high atomic resolutions, similar like crystallography. And it's a mix in between crystallography and electron microscopy. Okay, plenty of people are using micro-ED right now to analyze small compounds, small compounds, natural compounds. Even some people are using it as well for proteins. Okay, you still need crystals. So that can be the tricky part with micro-ED. Okay. So this is our life science TEM portfolio. I think probably everyone is already familiar here with this portfolio. Of course, today I will focus on the Glacios because that's the one you have here in campus. But of course, you can as well get access in Purdue University to the Cryos. It's the real state-of-the-art instrument, okay, for analyzing single particle, micro-ED, as well as tomography studies. So with that in mind, let's dig a bit more into the Glacios you have here. So of course, I'm guessing everyone here is already using the Glacios. Raise your hands. Who is using the Glacios currently here? Okay, not many people. So the rest of you, after this talk, I'm hoping I will motivate you to run to see this guy here, to schedule your time in the Glacios, please. Okay, the more the merrier. So what is the Glacios? So the Glacios is 200 keV X-spec microscope. Okay, what that means? That's the speed to which you are producing the electrons. So in general, the higher the speed, the higher the resolution you will be able to get. Okay. So the software, I will explain as well how the software works, what kind of software you can get in the Glacios and the upgrades you can do. Selectlist, that's the energy filter, and I will explain how it works. That's super important to increase the signal to noise ratio in electron microscopy. In electron microscopy, you have plenty of background, okay? And it's all white and black. It's not these beautiful, super resolution confocal images. It's nothing like that. It's all black and white. So you really need to increase as much as you can the signal versus the noise to be able to see your particles, or to be able to see, the particle of interest in the context of the cell or the tissue for tomography. So that is super important, and the Selectlist is doing an amazing job in increasing that, in doing increasing the signal, reducing the noise. And of course, the Falcon 4i, that's the new direct electron detector camera like we have. So it's not a CMOS camera, you are literally detecting individual electrons in your camera. So that means the quality of the image you can get is impressive, okay? So as I explained, combining all those three, right now, you have only the Glacios and the Falcon 4, it's not the Falcon 4i. So the Falcon 4i is a bit faster if you compare with the Falcon 4, but you are missing the middle part, the Selectlist. That Selectlist is very cool to have when you are doing single particle analysis studies, but it's essential to have if you want to do tomography studies, okay? And I will explain later why that's important. So of course, for the people that are already using the Glacios here, you know how it works. You have the grids. A grid is usually a 3 mm copper grid, usually. It's very tiny. So that's the one. Let me see if I can put here my pointer and you can see here. So this is the grid we are talking about, okay? And those grids are called auto grids, the ones going into the Glacios. Why? Because they are clip grids. So we have a C ring, like it's a copper ring. We put the grid in there, and after we put a little ring again over the grid. So all the three parts there, we call the auto grid, and that is super stable, okay? So those auto grids are going into this cassette, so you can load 12 different grids at the same time in your Glacios. So that's very cool and very convenient because you have a high throughput, so you can screen very quickly 12 different samples, 12 different conditions of the same sample, etc. So it's very cool. This cassette with the 12 different grids is going into this, like it's called the capsule or the nano cap, 2 different names for the same thing. So very confusing, guys, and we apologize for that. So this nano cap is going into your Glacios here, okay? You can collect data there. Again, the beauty of the auto grids is like, let's say you are collecting data in the Glacios and you have a 3 angstrom resolution, but you really want to push the resolution much better, So you can reuse the same grid, transfer that into your cryos in Purdue and collect data there. So it's a high connectivity without ice contamination in between Glacios and cryos. So that's something to keep in your mind, because having the capability here to screen grids, to have already a very good preliminary data set can help you a lot to transfer that into the cryos in Purdue later if you want to use that, to even increase better the resolution. Okay, so let's dig a bit more into the software like you can get in the Glacios. So we, put in the market very recently the smart EPU, software ecosystem. I don't think you have that here right now. You have probably only EPU system. So the smart EPU, how it works. So as I say, it's an ecosystem. Everything is run by Athena, okay? And you have 3 parts in this ecosystem. You have the data acquisition part, and this can be EPU, the version 3, that's the latest version, or this can be GPU multi-grid. And I will explain what are the differences in between GPU 3 and GPU multi-grid. That's for automated data collection. Okay, so automatically, once your grids are in the Glacios, you can start doing all the data collection. You don't need to be there all the time monitoring data collection. Another very nice thing is image evaluation, like it's part as well of the smart GPU. So this can be done with the thing that we call quality monitor, or you can even integrate CryoSpark Live. Probably some people already doing single particle analysis are familiar with CryoSpark. So we are right now integrating, it's a software to do all data analysis of your data, okay? And to get the 3D reconstruction at the end. So you can all have the quality monitor, like it's quite simple. It's only for CTF, motion correction, or you can get the CryoSpark like you can do everything there, on the fly. So when you are collecting data with GPU, automatically the microscope is transferring the data and doing all the processing. For example, picking the particles, doing 2D projections, classifications, initial volumes, etc., all in the same ecosystem, okay? And finally, we add in this smart GPU the thing that we call the smart plugins. And this is very cool because you can totally, I will say, tweak the way you collect the data. So for example, if the latest 10 images are not coming good, the focus is not good, you cannot see the particles, etc., you can automatically, the microscope can tweak that and can stop collecting data in that square and move to the next square. So we are ensuring you, like almost 100% of the data you are collecting is a good data. So you are not wasting time collecting data in a bad area of the grid, like after you need to put away during data processing. So with the smart GPU ecosystem, all the data you will be collecting, you will be able to use it later for data analysis and data processing, okay? So 3 parts: automated data collection, GPU or GPU multigrid, the quality monitor or CryoSpark Live, and the plugins, okay? So let me show you a bit more what is the GPU 3, okay, in the smart EPU ecosystem. So this is the typical, interface that you will be able to see when you open a EPU on the smart EPU ecosystem. So it's all with traffic lights. So it's very easy of use. Every direct alignments like you need to do on regular basis in your Glacios, everything will be done automatically by the microscope. If something is not working, you will have a message. It's going to become red. You click on the message and recover button, and everything is going to be done and resolved by the microscope itself. So this is super useful and convenient for people, like they don't have 15, 20 years of experience in cryo-EM, but they really want to start using the microscope. So everything is done automatically by the microscope. So it's very, very easy. Even my kids can do it. I don't trust them, but probably they can do it. I know, exaggerating. So another very cool thing that you have with the EPU 3 smart GPU version is that you can choose the experiment type you want to do. So you can do a grid screening if you are only planning to screen. You can do medium resolution, or you can do high resolution. So depending on the one experiment you are selecting, magnification, pixel size, etc., those parameters, like some people are a bit lost, especially at the beginning. What magnification I need to use? Oh my gosh. So everything is already decided for you by the microscope. Again, it's very versatile because, for example, expert users, like they feel more comfortable. Oh, I don't really like that magnification, like the microscope is choosing. I would prefer to do my own thing. You can change that. So it's very versatile. So for expert users, you can still play around with the microscope, no problem, and decide your own parameters. But for people like that, new in the technology, everything is already done. Okay. So this is the EPU multigrid, and there are different things. So you can get with GPU 3 version 3, that's the one I explained to you right now, or you can with the GPU multigrid. The GPU multigrid is automated data collection for single particle analysis, but you can increase dramatically your throughput. And let me go very quickly through this, because I think I have a video, like it's gonna be better to show you like 25,000 words. So let me try to play this video for you guys. It's playing, yes. So the thing, how it works. So you are loading into your Glacios all the 12 different grids, okay? And you are generating low magnification tile sets or atlas, like we call them, So this thing here is your grid. As I said before, the grid is a circular 3mm copper grid. Inside your grid, you have squares, and inside the squares, you have holes, and those are the ones you will be using for taking the data there, okay? So you are doing all those atlas automatically from one grid to the next, and you are screening the grids, and you realize, okay, over my 12 grids, only 3 are very good, and I want to collect on all those 3. So you can queue using GPU multigrid. you know, all the areas you want to collect in all those 3 grids. So even when you are done with one grid, the microscope automatically will change that grid and put the next one you want to collect on. So you can, with this GPU multigrid, you can keep collecting for days and days and days. You can go to Vegas, party time, guys, and the microscope will do everything for you guys. So it's very, very cool. So again, with GPU multigrid, you have a higher throughput, like with a regular GPU version. And I have some examples of, you know, customers of us and collaborators of us already applying GPU multigrid in their research. And this is, you know, this was published very recently, and this was done with a group in the UK. So we were able in 6 months from, you know, data collection to publication, 76 different structures in 6 months. So we isolate these filaments from patients, from brain patients, purification process, preparation of the grids, data screening, data collection, using all those parameters, and as you can see there, 8 hours for each data set. And at the end, in 6 months, from data preparation or sample preparation to publication only, 6 months for 76 structures. So, I mean, this is very impressive. When I start doing electron microscopy, can take me at the beginning, you know, the Paleolithic times, you know. So I was developing film, so I don't even tell you about all those stories, good memories with all the chemicals in the black rooms. But even when I had the first camera, it was a CMOS camera, and I was like already, oh my gosh, this is so nice. But can take you days and days and weeks to only collect the data, okay? And months to process the data. And you can see right now, 76 structures in less than 6 months. So it's very impressive. So as I say, and I'm highlighting here, you know, in the Glacios, you can install the GPU multigrid. You can do all the high throughput screening. You can do even preliminary data processing, you know, already coming from the Glacios. And after choose the best grids you really want to pursue it and get a very high resolution data on to the Cryos in Purdue. And again, very high connectivity. So the EPU quality monitor, so I explained to you EPU3, EPU multigrid for data collection. Right now, I will keep going in the circle. You remember the circle I present for a smart GPU. Right now, I'm going to the EPU quality monitor. So this is very cool if you really want to, on the fly, assess the quality of your data, okay? It's very simple because as you can see here in the interface, you can only see motion correction, CTF correction, you know, and the focus range. So it's very basic to see already the quality of your data is getting there. So for example, if every single image is coming with a CTF around probably 5-7 angstroms or maybe 10 angstroms, it's probably no good. Okay, you realize you need to do something with that. So it's already a preliminary assessment. If you really want to have 2D classifications, 3D initial volumes on the fly, you will need to install CryoSpark Live, integrated in the smart GPU ecosystem. Right now, people, like they don't have the integrated CryoSpark, they are still doing CryoSpark in external Linux box station, where you can transfer very quickly the data from your microscope to that Linux box, where you have CryoSpark Live and do already the data processing very quickly, okay? So there are different ways to do it. The smart plugins, and this is the last part of the circle. So this is very cool because we already have is all artificial intelligence machine learning, okay? And I know plenty of people are applying artificial intelligence right now everywhere, you know? So it's very cool because, for example, we have 3 main things like we already have integrated in the smart plugins. One is the smart focus. So use real-time CTF, you know, to autocorrect the microscope, the focus. So if something is not working there, the microscope will autocorrect itself to have the good focus you are looking for. Stage time, the same thing. So use real-time motion traces to adjust the stage there, you know, and to have the optimal setting, you know, for your data collection. And of course, the smart grid skip, that's the one I told you already before. For example, you are using the real-time CTF to really decide, this is a good area to keep collecting data, or I need to go to the next square. you know. So all those three are already implemented in the smart plugins, but we are working every 24/7 or R&D to even add more smart plugins here, different parameters, like you can, again, do everything automatically. And this is the thing as well. We are working with open, the thing that we call the application programming interface. And this is an open community system. I know some people are software people, they like to develop new things. So this is open for those kind of guys, like they can integrate their ideas, their softwares, their smart plugins in the same system. Okay, so it's not only Thermo working to develop that, but it's open to the rest of the community. So this is very cool. Okay, so talking about softwares, all the things I present until now, so all the smart EPU ecosystem is only for single particle analysis, okay? So it's a specific package that you will be able to use only for single particle analysis. What happens if you want to collect tomography data? In that case, you need to go with the tomography 5 software, okay? And this can be integrated in your Glacios if at some point you decide to go for tomography. So you know how it works, you have the automation, you can do the multi-site batch tomography, the cassette mapping, all the interactivity is there, so you can edit the tracking and imagine the regions you want, and you can as well do the advanced imaging, so those symmetric scheme as well, of course with the select list. So this is a bit Tomo Live, so you have the tomography 5 for data collection, and Tomo Live, similar a bit if you want to compare with the CryoSpark Live, you know, where you can on the fly start doing data processing with the tomograms coming from your Tomo 5 data collection. So it's very cool. So how it works, Tomo Live, you have the motion correction, stack cleaner, pair alignment, tomogram positioning, patch tracking, and reconstruction live. So again, if you want to compare with single particle analysis, it's going to be very similar to CryoSpark Live, okay? But for tomography purposes. So this is the thing. So Tomo 5 for data collection and Tomo Live for data processing on the fly. Before, we have only FFI. So this is called the fringe-free imaging, and it's a very technical thing in the cryos, okay? The very high level microscope, you know? I know some people here that they like cars. So I will say the cryos is probably a Lamborghini Maserati. We were talking about cars like last night, so I remember that. And maybe the Glacios is going to be already a very good Ferrari, okay? So before, we have already the FFI only on those Maseratis, you know, on the cryos. Right now, we are integrating FFI as well in the Glacios, you know, in all Ferrari. Why? Because you can really increase dramatically, you know, the throughput, okay? You can go, for example, right now, I know you have AFIS in your Glacios here. So it's about 400, 500 movies an hour, you know. If you integrate the AFIS and FFI, and this is with the Selectis and Falcon 4i, so it's a bit higher, you know, it's around 650 movies. So I'm guessing because you have only the Falcon 4 right now without the Falcon 4i, it's going to be probably between 400 you are collecting right now, probably to 550, 500, 550, you know, with the Falcon 4. But it's already a very good increase. Why? Because you can collect much more images per hole, you know. So that's very cool. Some questions here regarding software before I move to the select list and the camera. Everyone is good? So this is the select list. And you can see in the right side, that's the physical select list. So I will put a pointer here again. And you can see here, this is the select list, and this is the camera, you know, post energy filter. So how it works, this is the internal anatomy of a TEM, transmission electron microscope. So you are producing your electrons here in the field emission, you know, the FEC. So let's imagine, it's not totally correct, but let's imagine like all those electrons, they have all the same energy, okay? This is your sample here. When the electrons, and this is the cassette with all the 12 different grids. So when the electrons are reacting, you know, with your sample, some of these guys are inelastically scattered. And those are the guys like they are in red and yellow. Those are the noisy electrons like you don't want to have in your image because they are giving you all the noise, but not the signal, no information. So that's the energy filter here, how it works. Physically, you have an opening. It's a physical opening, like we call the slit, okay? And you are selecting, depending on the opening, bigger or less bigger, I will say, you are really being very picky in selecting only the electrons, like they are not inelastically scattered. Like they are the ones, like they don't have any noise, but they have all the signal you need. So that's the reason when you are using the select list, you are removing all these crappy electrons, like they are providing you with only noise. And you are really having the best electrons you can to have a very high quality of your image. So that's the reason the signal to noise ratio increased dramatically with the select list. Okay, and I will show you some examples here. So in this image, and this is in the Glacios, okay? This is in your machine. If you install the select list in your machine, what happened? This is the typical image that you can see without a select list. And I don't know if you can see that from far away, but here in my desktop is very clear. It's almost very difficult when you are working at very close to focus, to see the particles. Even working even closer to true focus, 0.38 microns is very close to true focus. And using a thin AB opening of the slit, I was explaining before, you can already see much better the particles, the proteins there, okay? So you are increasing dramatically the contrast. This select list energy filter is very stable. I don't know if some of you already have, familiarity with other energy filters in the market. When I was the director in NCI, I have another energy filter in the market because this guy wasn't available yet. And believe me, it was a pain in the neck because every single 2 hours, or even more often than that, I need to do all the tuning of that energy filter, okay? And that was no fun. Here you can see, you start the session, data collection, to do all the alignments, tuning of your select list, and you can keep going for hours and days without thinking about the select list anymore because it's super stable. Energy filters can be very picky with temperature variations in the room. This is like a rock, solid rock. The only differences and things that you can see here, those little bumps, are when automatically your Glacios is refilling liquid nitrogen in the machine because everything is under cryo conditions, okay? So every probably 7-8 hours, your Glacios is adding liquid nitrogen in the machine. So that's the reason you can see these little bumps in there. That's the only thing. Other than that, it's a rock. So let me explain right now to you the Falcon 4i. I know you have here the Falcon 4, and I put together a small comparison in between Falcon 4 and Falcon 4i. This is a relatively easy, and I will say probably not too expensive, upgrade to do in your machine. So you can go, for example, if you take a look at the DQE, it's an important value for direct electron detector cameras. We go from 0.2, 0.3 to 0.5 in this mode. Okay, this is a mode to where you collect data on the microscope. Let's take a look at the high throughput. Right now you have a 250 frames per second, like it's already quite good. You can get to 320 frames per second with the Falcon 4i. So you are collecting data faster, but not only that. The camera overhead, what that means, that's the speed to which you are transferring the data from your microscope to external Linux station or wherever. So right now takes 2.5 seconds versus only 0.5 seconds per image. So you are collecting data faster and you are transferring that data faster as well. So the throughput you are having is much better at the end. You can see here, for example, this is the Falcon 4i where you have a large pixel. All this gray area, this is the background, okay? All those little guys here, little mountains, are individual electrons, okay? So for cameras, other cameras, like they have small pixels, it's almost impossible to distinguish those individual electrons from the background noise. Very complicated. With our camera right now, you can really literally see how easy it is to distinguish all the 3 electrons individually from the background noise. So this again provides you with a high quality image at the end. How that translates to you guys. I don't know if, you know, some people here are crystallographers. Yeah, 1, 2, yes. So, okay, everyone is already very familiar with bifactors in crystallography. I was using bifactors forever when I was a crystallographer, but it's a value like it's becoming very, very important as well for cryo-EM, okay? The bifactor, what is that? It's a figure of merit for optical and detection quality. So the lower is better. Why? The lower the bifactor, the lower this number is, that means like the less number of particles you will need to collect to get to the same resolution. And this is a nice comparison. This is with the cryos. I apologize. I don't think I had this with the Glacios, but I can try to find it if you are interested. So this is our cryos, XFEC, Celestis X Falcon 4 versus another vendor camera. And you can see the differences in bifactors. This means, like, if I want to get to 1.9 angstrom resolution, I only need to collect 60K particles with my Celestis Falcon 4 cryos combination versus over 1 million particles with the cryos and other vendor camera. So again, high throughput. And you are here in a core facility. So the more high throughput you get, the better for everyone, because you will need to use less time in the microscope to be able to collect the number of particles you need to get to very nice resolutions. So the less time per project you are using in the microscope, the more projects you can allocate into that Glacios, okay? So it's very convenient for you because, again, high throughput, high quality data. Some questions regarding Celestis or Falcon 4i before I go for applications results. Yes?
How much does it cost?
That's that guy answering that question. It's not me.
It sounds expensive.
It's totally worth it. It's like driving a Maserati.
Sadly, our team is not here.
I hope you are recording this. I hope you are, because otherwise you can show that to the dean. You know, we have justification here, guys. No, but it's a good question, and we can take that offline. You know. Okay, so application results. Single particle first. I wasn't sure, you know, and I think I did well, because some people here are, I don't want to call you newbies, but they are not still using the Glacios yet. So how the single particle analysis work, the pipeline. So you have the sample preparation, like it's still, you know, biochemically speaking, you need to express and purify your protein of interest or micromolecular complex, put that into a cryo-EM grid, go to the Glacios or the cryos for data collection, and after 3D reconstruction to get the 3D volume of your protein at the end. So let me, you know, play this video. Hopefully it works. Yes. So all those little guys moving around, those are proteins, okay? And they will be vitrified at minus 180 degrees Celsius right now. So they are all in different orientations, okay? Almost native conditions. You don't have any artifacts there. That's your grid. That grid is going into the Glacios, transmission electron microscope, and you are collecting data. This is the typical image you will be able to get by cryo conditions of your protein. After that, computationally, everything is computational right now. You will be able to generate the thing that we call the 2D projections or 2D classes. So, you know, the more 2D projections, different orientations of your particle of interest you have, bottom views, side views, you know, top views, and in between, the more accurate you will be able to create the 3D volume, the 3D reconstruction at the end. So that's the reason we call it millions of, you know, particles. We align those particles together in the same orientation to extract later on all the information from those 2D projections into the 3D reconstruction at the end. I don't know if you already tried this, but the first time I saw my protein, like I was really working, I don't know, maybe for months and years on this when I was a postdoc at Scripps in San Diego, La Jolla. And when I saw it the first time in 3D, I was like, oh my gosh, this is almost the best day in my life. Because you realize it's real. It's not something like, mysterious anymore. That protein is real. You can play around with it, and it's super cool. So what kind of data? And again, I want to emphasize, it's always sample dependent, okay? Not every single sample will get as apoferritin to 1.6 angstroms. No way. I'm telling you that right now, honestly, it's not gonna happen. The majority of you guys I know are working with very difficult and challenging targets. So, it's gonna be more difficult for you to get to these kind of resolutions, but it's not because of the hardware. It's because of, biologically speaking, you have a limitation in your sample, okay? So that's the reason I wanted to show you, like the apoferritin, the one in the middle at 1.6 is the test control sample, like we always use for single particle analysis. It's a very relatively big sample. It's very robust and stable. It's not flexible or moving around. So that's the one like we use to realize what is the limit of the hardware we are having, okay? So we know with the Glacios, with the Celestis, this is with energy filter, and the Falcon 4i, we can get a 1.6 angstroms on apoferritin. We know that's the limit we can get with the hardware, okay? And after you have different examples, proteosomes after only 12 hours, 2.1. GABA receptor is only 210 kilodalton. It's a membrane protein, quite challenging, it's 2.4, after only 12 hours. GPCRs, another membrane protein, quite challenging again, 150 kilodalton, 2.6 in probably 20 hours, so less than a day. You have the CDK, it's a small enzyme, like it's only 85 kilodalton. To be honest with you, for single particle analysis standards, it's quite small. So we can get 2.5 angstroms in less than 20 hours, less than a day. And even hemoglobin, human hemoglobin is very small and it's very flexible, heterogeneous, everything we don't like in cryo-EM, So we can get a 3.4 angstroms in 20 hours as well. So again, depending on your sample, those resolutions can change, but this is to give you an idea of what you can really get with the Glacios, Celestis, and Falcon 4. And I wanted to show you this map. This is, as I say, the test control sample, 1.6 in, again, less than 9 hours of data collection. This is with the Celestis Falcon 4i. You can probably collect 9 hours data collection in this case, 400 movies an hour. So this is very similar to the things you are having here. And I put together all the, all the process for data processing. So motion, CTF correction, particle picking, 2D classification, 2D projections, 3D refinements, etc. And you need to be back and forth. I don't know if you're familiar with papers on cryo-EM. When you go to the supplemental figure, you have a huge diagram there from where you start until the end for the 3D volume. And it can be quite chaotic, so it's always back and forth. So you can see we collect plenty of data, and at the end, we use not that many data to get the 1.6, but it's very impressive. You can already start seeing what are molecules and other residues like this beautiful one in here. So it's very cool. So more bifactors, and I will go quite quickly on this. So we compare the same sample, there, with the same number of particles, and you can see without the energy filter, the bifactor is very high. So it's 2.3 angstroms. When you use the energy filter and you are closing that opening even more, so you are being very picky with the electrons you are selecting, you can see the bifactor is getting lower and the resolution is getting higher. So again, a smaller bifactor equals higher resolutions equals higher throughput. So something to keep in your mind for that select list. More examples here, again, the same idea, comparing bifactors with and without select list. So you can see you need less particles to be able to reach even better resolutions with the same sample. And again, this is a GPCR. You only need less than, a bit more than 200K to be able to get to 2.6. So it's very cool. This is another example as well that you can see. We collect data in the Glacios, and you can see, we can sell it, and we can even already visualize, the cystic Dalton bound protein of interest in this state. So it's very, very cool. Another very challenging sample. This is very difficult to crystallize. Plays a very important role in different diseases like, epilepsy. Is a target, for plenty of pharmaceutical companies, to create chronic inflammatory pain. And again, very difficult to crystallize. So Cryo-EM is not perfect yet, but I wanted to show you this because this is a very difficult target. It's not apoferritin, like everyone can do it, even again, my kids. This is a very difficult target. And we were able to get a micrograph image on this under cryo conditions, get the first, 2D classifications and start working on the 3D reconstruction. Still plenty of work to be done, but it's getting there. So it's very exciting. So cryo-TOMO, I would like to explain to you some results on cryo-TOMO. First thing, cryo-TOMO is a bit different, regarding how you prepare the sample. So you need more instrumentation and you have extra steps. So the first thing, we are working with cells, viruses, or even tissue directly, okay? So the first thing you need to do is, in this case, prepare your cells, vitrify your cells, do some light microscopy, to being able to localize where is the protein of interest in your cell, because the cells are very crowded. The cells are very thick as well. If you try to go from here to here, it's not gonna work. Everything will be black, okay? So the first thing you need to do is like the thing like we call the focus ion beam. So you need to open windows into your cell in a way like you can remove all the material that you don't want and create these thin lamellas in between 250 nanometers, like you can transfer to the transmission electron microscope to be able to collect tomography data. So that's an extra step and an extra instrument to be able to do this. And of course, when you add in the microscope, you can collect the data and do, all the data processing you need. So let me play this video for you guys so you can see a bit the difference. In this particular video, we are working with viruses. So the viruses are, purified, they are vitrified in the cryo-EM grid, again, minus 180 degrees Celsius. You are transferring that grid into the transmission electron microscope, can be the Glacios or the Cryos. And another big difference, if you compare with single particle analysis, is the way you are collecting the data. So usually you collect the data in single particle analysis with no tilt. In tomography, you are tilting the stage inside the microscope. So you are generating a tilt series, like we call, it's like a movie, like we call the tomograph. Usually it's minus 60 or minus 70 degrees to plus 60, plus 70 degrees. And the intervals in between those is usually 3 to 2 degrees, okay? So when you put all those series together, that's the thing that we call the tomogram. Again, after that, computationally, you can do tomogram, subtomogram averaging, and segmentation work using different softwares to be able to get the 3D volume of your, in this case, is the virus, but you can get as well the 3D volume of your protein of interest in the context of the cell and the context of the tissue. So again, it's very cool because it's all in situ work. As I said before, you can do cryo-TOMO with proteins, viruses, bacteria, and cells. But because the cells are very thick, when you are working with materials like they are thicker than 300 nanometers, you will need to do the cryo-FIB cell. You don't have any choice. If you are working with bacteria or viruses like they are less than 300 nanometers, you can go directly to the transmission electron microscope and do the tomography. But in the case of the cells or tissues, of course, obviously they are thicker than 300 nanometers. So you will need to go with the cryo-FIB. So very quickly, those are the 3 machines like we currently have in the market for cryo-FIB. So we have the Glacios, like it's a dedicated machine for cellular cryo-electron tomography sample prep. It's all cryo, preparing the lamellas for cryo samples. The FIB is, a gallium source. For the plasma FIB, the Hydra in the middle, that can do cryo and can do room temperature as well. So for example, if you are interested in doing volume scope, volume EM at room temperature, you can do that in the Hydra. You cannot do that in the Aquilus, okay? So you have much more versatility and flexibility for applications in the Hydra versus the Aquilus. Another huge difference in between Hydra and Aquilus is the plasma. In this case, the Aquilus, as I said, is only gallium. In the case of the Hydra, it's a plasma FIP. So you have 4 different ion species, like xenon, argon, oxygen, and nitrogen, that you can switch very easily in between one to the next. It's only 10 minutes to switch the species. And depending on the sample you are working with, for example, if you are working with resin embedded samples, we are realizing like oxygen is working much better than gallium. You have much less curtain, you have much less charging, you have much less artifacts. Okay, so that's super cool. So something to keep in your mind. In both machines, Aquilus and Hydra, you can get an integrated fluorescence light microscope. And this is very cool because you will be able to do CLEM inside the machine. As I said before, the cell is very crowded. You have plenty of things there. So you need to be able to target with a fluorophore the protein of interest. If you go to the machines, Hydra or Aquilus, to prepare the lamella, you want to be confident, like where you are preparing the lamella is where the region of interest is still located. You don't want to be blind making the lamellas without knowing if you are really targeting your protein of interest. So that's the reason we are integrated fluorescence light microscope, where you can switch back and forth in between milling and coming back to the microscopy, to the light microscopy, to see if you really have the protein of interest still there in your lamella. So that's very convenient. And of course, the ARTIS that was launched very recently, and it's a, I will say, very high throughput machine to prepare cryolamellas. So it's all cryo, and it's a mix in between the Hydra and the Aquilus, in the sense, like the ARTIS, you have 3 plasma ion species. So you have the xenon, argon, and oxygen. So it's a plasma FIP, and but it's only for cryo lamella preparation. But the ARTIS has the autoloader. So you can, similar to the Glacios, so you can load 12 different grids there and prepare lamellas automatically from all the 12. So very high throughput for lamella preparation. You have as well the integrated fluorescence light microscope there, and the beauty of the ARTIS versus the Hydra on the Aquilus is like in the Hydra and the Aquilus, you don't have the 3-point coincidence points. So you have the iCEM, you know, the E column for imaging, you have the E column for milling, and you have the integrated fluorescence light microscope. And all the 3 cannot be a coincidence because we don't have enough space and it's complicated. In the ARTIS, all the 3, you know, the iCEM and the E column and the integrated fluorescence light microscope, they are a coincidence. So that's very, very convenient for you guys. So again, all the 3 instruments we have for the CryoFit. I already explained this. These are capabilities of the Aquilus and Hydra. Cryolamella automation, you know, you can do the integrated fluorescence microscope, the cryo lift out. I will go very quickly through this. This is very important for tissues. And of course, the cryo volume imaging is only for the Hydra. Room temperature or cryo volume imaging, only on Hydra. So I already explained to you with my hands, you know, you have the iCEM column for imaging, the FIP column can be gallium or can be the plasma FIP. And, you know, the integrated fluorescence light microscope is not in this image, but it's on the other side. And this is the Hydra, you know, again, you can see how it works. You can switch in between, you have an interface, you know, where you can decide which ion species you want to use, and you can switch in between those very quickly, less than 10 minutes. And I will show you very quickly in this video how you prepare the sample. So this is the typical data you will be able to get using cells, you know, or tissues when you are doing all the segmentation work. So you can see all the organelles there. It's very impressive. This is your grid. So your cells are growing up directly on your grid. Okay. When the cells are happy in the grid, you will be able, you know, to vitrify them using the VitroBot. It's the same machine you will be using for single particle and for tomography. It's the same machine. So those are your grid with your cells there. So you need to create a very thin layer of, you know, buffer around those cells, okay? And you are doing that with the blotting like you saw. in the video. So now you have this very thin aqueous film around the cell. You are plunging very quickly into that cup, like it's eaten at minus 180 degrees Celsius, and you are transferring that grid with your cells into the aqueous or the hydra or the artist to prepare the lamella. And let's see how it works for the lamella. I have another video here I would like to show you. This is specifically for the aqueous. So if you go internally to visualize the aqueous, how it works, this is the solder, it's under cryo conditions. So you have two different grids at the same time in the same solder, okay? So first of all, you have a protective coating, to protect your cells and your sample there. And after you will be zooming in one specific grid, and you will be able to localize, using the iCEM. So this is the electron beam for, visualization and imaging, being able to visualize, where is the cell you want to target. So this is your cell in your grid, okay? Everything under cryo conditions, don't forget. And after that, you will be able automatically, with the software we have in the aqueous and the hydra, to select the areas in yellow that you want to mill away. And in the middle is the lamella. That's the one you want to save to transfer to the transmission electron microscopy later. So this is the focus ion beam, milling away all that material that you don't need, opening those windows into your cell. And after that, you will be transferring that lamella into your microscope. This is the program that you will be able to see, in the aqueous or the hydra to do automatically create different lamellas in your grid. So as I said before, this is your grid. So you can see different, cells in the grid. Depending on what kind of cells you are working with, you can get 20 cells in a grid or 15 cells in a grid, depending. So you select the areas that you want to prepare the lamella there, and automatically, the system will prepare those lamellas in the grid. So in a grid, you can get 12 to 15 lamellas ready to go to the transmission electron microscope. So it's very, very cool. And those are the lamellas, like they are already done. Can you see all those different sides? Those one lamella, over lamella, over lamella. So all those are the ones that you will be able to use in your transmission electron microscope. Okay. So this is the integrated fluorescence microscope, and you will be able, to see. It's very cool because you can correlate both. So you can get the iCEM image and you can correlate with your fluorescence, and you will be able to really target. Okay, I want that area. I want to really, mill and prepare the lamella in that area. So, and after, when you are preparing the lamella, you can come back and use the integrated fluorescence light microscope again to double check before transferring that lamella into the TEM, that you still have the area of interest there. You didn't lose it. It's still there. So you are super happy. You transfer that lamella into the microscope and you start collecting data. So this is the typical vision here. You can see the iCEM, the TEM of the lamella, the tomogram you can obtain. And this is the average of thickness of those lamellas. The thin the lamella, or the thinnest the lamella, the better for you guys, because you can get better contrast. Okay. Again, don't go too crazy either, because the thinner the lamella, the more fragile the lamella is as well. So usually people right now are working in between 115 nanometers, that's the bit limit, and 250 nanometers. Okay, so that's probably the range that people are working with. Lift out. This is super, super cool technique. And this can be done not in the artist. We don't have lift out in the artist, so you cannot do that in the artist, but you can do lift out in the aqueous and the hydra. Okay, this is very cool for tissues. Tissues, I don't know if you're familiar, it's very thick. You need to do the high pressure freezing, in specific planchettes. You cannot do that with a vitro because it's too thick. Okay, so as you can see here, is the planchette. So those planchettes are compatible with the aqueous and the hydra. So you can transfer that into your machine, select a chunk of material that you want to mill. You take it away with this special needle, and this chunk of material is being, I will say probably a bind or glue, kind of a glue thing, into a special grid. This is the special grid, like we call the half moon grid. Okay, so you put your chunk of material there, and you start literally milling away material directly on the grid, on that special grid. So that's very cool because after, again, with the integrated fluorescence light microscope, you can double check like your region of interest is still in that lamella. So again, select the chunk you want to pick up. A special needle like will glue to that chunk of material. That chunk of material will be glued to the special grid. And after one that is glued there, you can start the milling process to make the lamella there. So it's very, very cool. So we already know how it works for tomography data. So I will pass that very quickly. And because of timing, I will go, this is some samples like I wanted to show you, coming from the cryos. So this is the typical tomograms that you can get, after some data processing and doing segmentation work, you will be able to see, all the videos and everything in 3D. More examples, this is very cool. I need to play this video because otherwise I won't sleep well tonight if I don't show you this video. So this is very cool. This is a skeletal sarcomere and cardiac sarcomere, all by tomography in situ work. So you can see already skeletal actomyosin and cardiac at 6.6 and 7.7 angstroms. And if you really go into the core of that proteins, you can see it's even higher resolution, 4.5 angstroms. You can see how the nebulin and the actin are binding together. And even you can already start to localize some very important residues, like are playing an important role in this particular binding. So don't forget, it's all in situ work, so the resolution is quite impressive. Virus, again, it's HIV virus, proteins, particles. So we were able to get 4.2 angstroms with only 13 tomograms. So very impressive. Another example here, even higher resolution, 3.8 with only 8 tomograms. This is everything on the cryos, okay? Cryos, Celestris, and Falcon 4i. So this is the kind of resolutions you can get working with the cryos. Again, sample depending. It's always sample depending here. Let me show you some examples with the Glacios. So some people are more interested in doing as well tomography at 200 kV. So this is the thing we have here. This is a proof of concept like this can be done. This is apoferritin on the Glacios. And I have other examples. This is a bacterial cell on the Glacios. This is a tomogram we collected. And this is, all the segmentation and work like we did after tomograms with tomogram averaging. Because the bacterial cell is less than 300, probably 300 nanometers in thickness, you don't need to do the cryo feed. You can directly freeze the bacterial cell into your grid and go to the transmission electron microscope, collect tomography data, do all the processing and get the data. So it's very cool. And this is coming from the Glacios, don't forget. More examples from the Glacios, again, 200 kV. This is a lamella that was preparing the aquilus from Chlamydomonas. And after data collection, you can see here, we were able to do all the tomograms with tomogram averaging and segmentation work. And you will be able to see all those beautiful organelles, ER, Golgi, and all those yellow things are ribosomes. So it's very, very cool. So again, all work in situ, high quality. This is all the work we did, with all these people. So we call the second resolution revolution because we had the first resolution revolution with single particle analysis in 2020 when we get the first real atomic resolution at 1.2 with apoferritin. Right now we are having the second resolution revolution with cryo-TEM. So it's very exciting. And I don't know if I want to spend, because I am already over time, if you are very interested in micro-ED, I can go very quickly on this. Otherwise, I will open the floor for questions. Your choice, guys.
I think that
let's do it. I like that. Okay, so very quickly, because I apologize. I'm from Spain. I like talking and I'm passionate about this. So I know I am over time. So micro-ED complements, cryo-TEM and cryo-EM single particle in the sense like you still need crystals, but you can get very high resolutions. So how it works? You prepare the crystals. Usually when you are working with small compounds, natural compounds, they are quite easily to crystallize. But you are working with very small crystals in between 200 and 500 nanometers. So they are very tiny to go for the typical synchrotron. It's not going to work. Okay. So those crystals, you vitrify them the same way in a cryo-EM grid. You go to the transmission electron microscope, you localize the crystal, you do a diffraction screening to see if you have some diffraction spots. And after you start collecting, diffraction till series continuously, okay, to generate, the phasing, reframing, and get the 3D reconstruction. So usually you need around, I will say on average, probably 3 to 5 minutes per crystal to be collected under micro-ED conditions. And depending on your sample, but on average, probably 15 to 10 crystals of the same sample are more than enough to get all the information you will need to create a 3D reconstruction at the end, okay? So if you compare with the other techniques, typical crystallography, you need large crystals, usually 5 microns by 5 microns, and sometimes they are very difficult to get those crystals. So the XFEL is another technique, like you can use small crystals, but sometimes it's difficult to access, this technique, and you need plenty of those crystals. We are talking about 83,000, so it's a lot. Micro-ED is a good compromise because you can use small crystals. You don't need a lot of those crystals, and it's in-house technique because you can use the transmission electron microscope like you already have here, as long as you have the micro-ED package, to do micro-ED. So this is the micro-ED package, it's a special GPU-D, it's a special software to be able to collect data with a continuous tilt in your microscope for micro-ED. And of course, you need as well a special camera, like we call the Zeta-D for diffraction. Like it's designed specifically to have a very good resolution under diffraction mode. I wanted to show you some examples of, already proteins and products that have been resolved using microelectron diffraction. And as I said before, you have different examples here. And I would like to pass to the next one to show you this. I like this comparison in between crystallography, XFEL, and micro-ED. This is the same protein, so it's a granulin, like it's only 29 kilodalton, so it's a very small protein, too small for single particle analysis. So if you do crystallography, you need 5 microns by 5 microns crystals, and you need 21 crystals of those big crystals, You get 1.7 angstroms. It's still the best resolution, okay? XFEL, you can use small crystals, but you need over 80K. That's crazy. I mean, who get 80K crystals? I mean, it's not easy. And you get 2 angstroms, okay. Micro-ED, I think, even if right now is the technique like provides you with less resolution, it's only 2.7 or 2.8. For me, it's a good balance because you are only using 5 tiny crystals, only 5. And it's an in-house technique because again, you don't need to go to the synchrotron or send your samples there. You can do everything here in your Glacios. So again, very good balance. And with that in mind, I am done. So thank you for your patience. I am 10 minutes over your time. So plenty of information. So I know it's overwhelming. I have my email address there. So take note of that if you need anything. And I open the door right now, floor for questions. Yes. Thank you, guys.
Those grids, right? So the grids that you showed where you showed the picture of the cells or the cartoon of the cells sitting on it. So we buy the grids and we put it on the cell culture dish and we grow the cells on it?
Yes, that's correct. But be careful because the majority of the people are using copper grids for single particle analysis. Copper is toxic for the cells. So you need to use gold grids or other substrates, like they are not toxic for your cells.
Okay.
And that's the bottleneck of all the pipeline I saw you, is how to make those cells happy in your grid.
Yeah. And then on top of it, they also treat the cells.
Yeah. So that's still the bottleneck of all the cryotomography techniques. Some people are struggling, I will say, to make those cells happy. Once you have the recipe and you are using mostly all the time the same kind of cells and you know how they are happy, you are golden. Because once they are growing there, the vitrification process, the preparation of the lamella, that is heavily automated right now, so you can make all those recipes quite easily. Data collection, data processing, again, the bottleneck is mostly, growing those cells happily in the grid.
So,
Yeah, of course.
So when you say grids, are they really holes?
Yeah. Well, for the there are holes, but for the cells you have a carbon coat in that grid, so the cell is not going into the hole. Yeah.
There's no mechanical.
Yes, that's correct. Yeah. Yes.
Question in the chat.
Oh, yeah, good. Thank you. How do you compare EM protein structures to alpha-fold protein? Oh, that's very good question. I was expecting that one. So AlphaFold is a very good thing for protein prediction. Okay, it's a protein prediction tool. But if you really want to understand the protein structure, you always need to have, experimental data. Right now, we are seeing, like, if you compare the AlphaFold prediction with your experimental data, it's very high quality, it's very, very accurate, but the experimental data never will go away. You know what I mean? Because you always need to have that to compare with the prediction. Because AlphaFold is always a prediction. It's never be the real structure at the end. So I hope I answered your question, Con.
Serious limitation. It's not considered.
I can add, yeah.
Yeah,
Everything's a monster.
yeah, yeah. And right now they have Alpha, totally. And right now they have the AlphaFold 2, like they are trying to resolve all those, bugs, I will say, but I completely agree with you. It's still a prediction, Yeah.
Yeah, so AlphaFold is a prediction tool that predicts structures based on previously determined structures. So you have a protein fold that had never been solved before, then AlphaFold is likely not to work. More importantly is that a lot of people now solve structures looking at inhibitor compound drug interaction with your protein or target. There's no way that AlphaFold can predict where those drugs will bind without having the actual structures. So maybe I'm biased, but we always rather solve structure than do prediction. We do a lot of predictions too, but for different reasons.
I have a question regarding iCEM.
Yes.
I mean, can you detect the flexible regions or disorder?
So I will repeat the question for the people virtually. So the question here is what happens when you have a protein like have flexible regions for single particle analysis. So the answer will be that's tricky because in cryo-EM, single particle is very similar to crystallography in that regard. We don't like that much heterogeneity. We don't like that much flexible regions because it's going to be very difficult to align those regions when you are doing the 2D classes and the analysis. So one solution, you can do several solutions. One can be try to stabilize that region, maybe with a FIB or maybe with another, protein, like can bind to that region if you can find it to stabilize that area. Other thing is mass spectrometry. People are using plenty of native mass spectrometry, can be cross-linked mass spectrometry as well, or even ECX mass spectrometry to try to understand, what happened with those flexible regions and try to, correlate, the cryo-EM map you are having with the mass spectrometry data you are having. So there are different approaches that you can try to get, a 3D volume of that region. Like it's going to be challenging only by cryo-EM. Yeah.
Yeah, just a quick follow-up. How do you know for sure your EM images is covered the whole molecule or only part of the molecule?
That's a very good question. So for the audience virtually, so how we are sure, like we are really, the 3D volume we are having is covering all the protein. So that's the reason I explained during the video at the beginning, like it's important for you to have as many orientations as you can of your protein. Because one typical problem like can happen in single particle is preferred orientations. So for example, you have all your proteins in the grid in only bottom views. So when you are doing data processing, you are missing plenty of information from the other orientations. So the model you will generate is not going to be accurate because you have only information from only one orientation. So that's the reason the more orientations you can get, of your protein, like they are in different orientations on the grid, the best, or I would say the best model 3D at the end and more accurate you will be able to get. Yes, that's very important.
Thank you.
Yeah, of course. Okay, guys. I don't know. Another question here. Okay, nice. Krishna, how do you prepare the samples for micro-ED? Is the same way as grid preparation for single particle cryo-EM? So very good question, Krishna. So for micro-ED, the first thing you need to do is to have crystals, okay? And again, that can be a bit tricky sometimes, but once you have your crystals, those crystals can be maybe too big for micro-EV, sorry. So people are using cryo-feed milling to mill those crystals and making it smaller in between 200 to 500 nanometers in size. Once you have those crystals homogeneous to the same size, you will be able to have a solution of crystals that you will be able to apply 3 to 4 microliters in a cryo-EM grid, and after vitrify that the same way you are doing in the vitro work, the same way you are doing for TOMO or for single particle. So that's the same way. But the tricky part right now is having those crystals, because you need to have the crystals, and having the perfect size for those crystals. Because sometimes, again, they are too big they are too small for crystallography, they are too big for micro-EV. So you need to be able to mill those crystals with a cryo-feed milling. Yeah, I hope I answered your question. Thank you. I don't think I have anything else there. No. Thank you so much. Yeah, thank you for coming.